Hello friends, how are you all? Hope so everybody is fine and healthy. Welcome to the today's session phylum Basidiomycota. In our previous 5 to 6 classes, we learned about the phylum Ascomycota. So in today's session, we learn about some important characteristics of phylum Basidiomycota. Basidiomycota means we have to remember the mushrooms, bracket fungi, rust and smut and puffballs. All the important fungi are placed under the phylum Basidiomycota. Everybody know about the mushrooms. From our childhood we used to familiar with the mushrooms. So these important mushrooms placed under the phylum Basidiomycota. So let us move to the main content. Coming to the introduction, the Basidiomycota, how the name derived for the phylum Basidiomycota and how the members evolved in Basidiomycota. The members of subdivision Basidiomycotina, when we are mentioning about as a subdivision, then we will end with the Mycotina. If we are mentioning the phylum, it will end with the Mycota. This classification I mentioned in my previous videos. So if we'll Basidiomycotina, these are highly advanced fungi compared to Ascomycota and to other fungi. These are highly advanced fungi, and the name Basidiomycota, why they given? Because the fungi produces sexual spores on a special club-shaped fruiting body. That fruiting body is known as basidium, and these sexual spores produced are called basidiospores. These basidiospores, how they are produced means, here you can see in this picture, this is the basidium, which is a club shaped structure on which basidiospores are produced, and they attach to the basidium through specialized structures known as pterygmata. These terms we have to remember for examination point of view. And fungi belonging to this subdivision Basidiomycotina or phylum Basidiomycota, these are generally referred as club fungi. This one we have to remember. So why the name Basidiomycota is given to the fungi placed under the subdivision means, because they produced a club shaped fruiting body. That fruiting body is known as basidium, on which basidiospores are produced. Usually 4 basidiospores are produced, and these basidiospores are connected to the basidium through the sterigmata. That's why the name Basidiomycota. And most of the members included in this Basidiomycota are umbrella shaped mushrooms and toadstools, rust and smuts. These fungi evolved along with our agriculture, and shelf and bracket fungi, puffballs, sting horns and jelly fungi. Here you can see different type of fungi. Bracket fungi means they used to produce the fruiting body in the form of bracket, hence the bracket fungi the name was given. So these are the important fungi placed under phylum Basidiomycota. Coming to the general characteristics, as already we know that Basidiomycota fungi, they produce the sexual spores. That spores are known as basidiospores on outside of the specialized spore producing structure. That structure is known as basidium. And how many spores are produced? Usually it is 4 basidiospores on pointed projections, already told, that is known as pterygmata. And this basidiospores is nothing but sexual spores, so they used to be haploid in nature and uninucleate. Uninucleate means they used to have the single nuclei. And how these sexual spores are produced, just remember the Ascomycota, how the ascospores are produced, because of plasmogamy, karyogamy and meiosis. In the similar way, these basidiospores are also produced because of the three processes: plasmogamy followed by karyogamy and meiosis. And dikaryotic phase dominates the life cycle in the Basidiomycota. Dikaryotic phase. Dikaryotic phase means in the mycelium, each cell is having two nuclei. The name itself indicating karyon means nuclei, di means two nuclei. So two nucleate mycelium that dominates the life cycle in the Basidiomycota. And some other special structures are also present in the Basidiomycota, that is clamp connections and Dolipore septum. Each one is having its special functions. So let us see in the next slides what are those functions performed by these clamp connections and Dolipore septum. And absence of motile spores. So in this fungi, motile spores are absent. Motile spores means the spores which are having flagellate structures for moving. So that motile spores are absent in this Basidiomycota. That is presence of club shaped fruiting body known as basidium and production of 4 basidiospores which are attached to the basidium through the structure stigmata, and production of haploid uninucleate basidiospores, results of plasmogamy, karyogamy and meiosis, and domination of life cycle by the dikaryotic phase, and presence of clamp connections and Dolipore septum, absence of motile spores. These are the general characteristics. Coming to the somatic structure. Somatic structure is nothing but mycelium or thallus. So this mycelium consists of well developed septate mycelium. Septate mycelium means all we know that if it is a mycelium, this is the septation used to be there. And this mycelium is divided into individual cells through the septations. And here one specialized very important characteristics of phylum Basidiomycota is mycelium passes through 3 distinct stages before the completion of life cycle. Those 3 distinct stages are primary mycelium, secondary mycelium and tertiary mycelium. What is primary mycelium and why we are calling it as a primary mycelium? Primary mycelium is also known as homokaryon or monokaryotic mycelium. Monokaryotic mycelium means mono. Mono means single. Mono means single, karyotic means nucleus. It means this primary mycelium is having single nucleus. If you will take it as a single suppose if you take it as a primary mycelium, it is having single nucleus. Here you can see like here like that, each cell is having one single nucleus, and it consists of hyphae with uninucleate cells and usually develops from germination of basidiospores. This basidiospore, if it is basidiospore, it will give rise to the germ tube through which the primary mycelium develops. So primary mycelium generally develops through the germination of basidiospores. Initially, when basidiospore germinate, it gives rise to multinucleate mycelium in the initial stages. But later, septa laid down and it divides the mycelium as a uninucleate primary mycelium. That is, multinucleate stage is short-lived because the septa are formed and dividing the mycelium into uninucleate cells. That means primary mycelium is monokaryotic in nature and it consists of hyphae with the uninucleate cells and develops from the germination of basidiospores, and initially it is a multinucleate when the basidiospore give rise to the germ tube and it gives the mycelium, but later septa are laid down giving rise to the uninucleate cells. This is the primary mycelium. Secondary hyphae. Secondary mycelium means it is dikaryotic or dikaryon. That means which is having 2 nuclei in each single cell. And how the secondary mycelium originates means it because of the primary mycelium and its cells they are fused. That is somatogamy takes place and results in the secondary mycelium, which is n plus n status. It means here karyogamy and meiosis do not take place, only the plasmogamy takes place, that is cytoplasmic contents they will fuse but the nuclei remain separate, they do not fuse, hence the dikaryotic mycelium. We call secondary mycelium as a dikaryotic mycelium, and this dikaryotic mycelium or dikaryotic stage, it dominates the life cycle, means it will long lasting. This mycelium is long lasting, unlike Ascomycotina. In Ascomycotina, the dikaryotic mycelium, the dikaryotic stage is short lived, but in case of Basidiomycotina, it is long lived and it exists during major part of the life cycle, and this stage is independent extensive phase. Unlike the short dikaryotic phase of Ascomycotina, we already saw the dikaryotic phase in Ascus development, where Ascus mother cell develops and there dikaryotic phase exists. And that special important character of the secondary mycelium is this is associated with the special structure known as clamp connections, and through which dicaryotization takes place. Means how the dicaryotization takes place means through the clamp connections. I will explain in the coming slides about this clamp connection and how they will help in formation of dicaryotization. And what is dicaryotization? This is a process by which monokaryotic primary mycelium is converted into dikaryotic secondary mycelium. And to the tertiary mycelium, there is no much difference between the secondary and the tertiary mycelium. However, tertiary mycelium cell walls are very thick in nature, and this results in the formation of fruiting bodies called sporophores or basidiocords. This is the tertiary mycelium. So there are three types of mycelium in the Basidiomycota: primary mycelium, secondary mycelium and tertiary mycelium. Primary mycelium is uninucleate in nature and secondary mycelium is dikaryotic in nature, and tertiary mycelium is a binucleate mycelium which is organized into specialized tissues resulting in the formation of fruiting bodies. That fruiting bodies known as sporophores or basidiocords. This is the tertiary mycelium. So in a pinpoint, let us see what is primary hyphae means. It develops from a germinating basidiospore and nuclear status is N, and secondary hyphae results from fusion of two primary hyphae and yields N plus N cell, that is dikaryotic cell, with continues to grow as a N plus N hyphae. And tertiary hyphae exactly the same as secondary hyphae N plus N. However, it has thick walls that enable production of fleshy and wood sporophores, that is fruiting bodies, basidiocorp. This is the about mycelium in Basidiomycota. And dikaryon, I will explain about this dikaryon. Dikaryon is nothing but secondary mycelium, and how it takes place. Simply just we will revise once again. Most of the Basidiomycetes are heterothallic, that means they are having different thalli, and haploid basidiospore germinate to produce haploid mycelium, that is we called it as a primary mycelium. And primary mycelium, how it contains single nucleus, and each compartment called monokaryon because each cell is having only single nucleus, that's why we call it as a monokaryon. And plasmogamy occurs shortly after basidiospore germination. Because of plasmogamy, what will going to happen? It will give rise to the secondary mycelium. Plasmogamy occurs between two compatible hyphae and results in the secondary mycelium, that is dikaryon. So after plasmogamy, what happens? The nuclei migrates into monokaryotic cells to establish a dikaryotic condition. So this is the only plasmogamy. Plasmogamy results in the formation of secondary mycelium, that is dikaryon. This is the dikaryon, and this is also dikaryon, but this dikaryon is happening in secondary hyphae and tertiary hyphae. Coming to the clamp connections, the clamp connections are where they are present. They are present in the secondary mycelium stage. And what is the special function of these clamp connections? What is the necessity for having the clamp connections? So let us see. Clamp connections look like they are hook-like structures formed laterally in between dividing nuclei in the dikaryotic hyphae. If you will take this is the dikaryotic hyphae, it is because this is the single cell having two nuclei A and B, because most of the these Basidiomycetes are heterothallic in nature, that is having different nuclei. When they fuse and form the secondary mycelium, these nuclei are also different. So here let us give some name to those nuclei. Here we given the A and B, or as you wish 1 and 2. So where the clamp connection will form, clamp connection exactly forms between these two nuclei. It means here it will form. So it acts as a bypass for the nuclei. It helps in the movement of the nuclei, that is bypass for moving the nuclei, as they cannot pass through the septal pore, that is Dolipore septum. Dolipore septum I will explain in the coming slides. Dolipore septum will not allow the movement of the nuclei. So nuclei has to multiply and they has to form daughter cells. Then how it will happen? So through the clamp connections. So here you can see clamp connections. It formed here in between these two nuclei, and it hook like structure. What will happen while dividing the nuclei? This B nuclei, it will migrate to this clamp connection, and A nuclei, it will divide, and the new nuclei whatever formed, it will come close to this clamp connection base, and here B nuclei also it will divide, and whatever daughter nuclei that will come near to this A nuclei, that is newly formed nuclei. So this newly formed nuclei, they will migrate into the separate cell. Instead of migration, here septation will be there. That septum will form, so that here two septa, one is to cut the clamp connection and another to separate the newly formed daughter nuclei. Here these are newly formed daughter nuclei or newly formed two cells. Here you can see here septation is there and clamp connection also closed, and again two cells are formed, that is two newly formed cells. Again the same process will repeat. Here clamp connection will form, here also clamp connection will form, and this process continues like this. So how the development of clamp connection, whatever I explained previously in that picture, here I will show point by point. So when a binucleate cell is ready to divide, small lateral branch, that is known as clamp connection, arises from the cell between two nuclei. Already I told it will arise between the two nuclei, that is A and B, and begins to form a curved hook. Here it is a curved hook. Then the two nuclei divide simultaneously. So they are dividing at the same time. This everything happening in the secondary mycelium. We have to remember this point. This entire process is going to happen in the secondary mycelium. And one division orients obliquely so that one daughter nuclei, that is B, forms in the clamp connection, and another daughter nuclei B1 forms in the dividing cell. Here one nuclei it moved to the clamp connections excuse me, and another it forms in the cell. Here you see formation of daughter nuclei, and this clamp connection it will attach to the hyphae, and second division orient itself along the length of the dividing cell, so the daughter nuclei A forms near one end of the cell and other A1 approaches the nucleus B1 of the first division near the other end of the cell. So in the meantime, clamp bends over and it's free and fuses with the cell. Here you can see the clamp will fuses and with the this dikaryotic cell, that is this hyphal cell, it will fuses, then it forms a bridge or septum, so that daughter nuclei B passes to the other end of the cell and approaches the daughter nucleus A. So they form into a separate cells. So here it came into one cell, and here this newly formed nucleus came into another cell, and a septum is formed to close the clamp at the point of origin, that is point of origin is here. So here clamp will form. Here you can see this point of origin, clamp will be formed. Then another divide the parent cell into two daughter cells with A and B in one daughter cell, and another nuclei A1 and B1 in another cell. So this one daughter nuclei here and another daughter nuclei, and this process remain continuous. So again clamp connection is formed here, again clamp connection is formed here, again it multiply, again new nucleus forms, and this process continues. Clamp remains permanently attached to the hyphae. So presence of the clamp on the hyphae, it indicates that it is a secondary mycelium and it is a dikaryotic mycelium. Here we can see the complete picture of the clamp connections, formation of hook-like structure, and where the movement of the nucleus into this clamp connection, and division of nuclei simultaneously, and formation of septa to divide the newly formed nuclei into two different cells. Here the septal pore is surrounded by a cup-like or dome-shaped structure that is known as parenthosome, also known as septal pore cap or nuclear pore cap, and it is made up of a double membrane, and its function is to shut the pore. Why Dolipore septum, what it will do? Just it will close the pore, but it will allow the movement of continuity of cytoplasm, but it will not allow the movement of nuclei. That is the function of this Dolipore septum. So what is the main function of Dolipore septum means it will not allow the movement of nuclei in the hyphae but maintains continuity of cytoplasm. Here you can see the Dolipore septum. If you take, this is the mycelium, this is the septum. Here central pore, it will swells, and then here this barrel shaped structure formed. That barrel shaped structure, it will form like this, and this barrel shaped structure is known as parenthosome or septal pore cap, we know. And these terms we have to remember in examination point of view. And this main purpose of Dolipore septum is to stop the movement of nuclei while maintaining the continuity of cytoplasm. And coming to the asexual reproduction, all we know that it takes place by means of budding. Budding means conidia formation and fragmentation of hyphae, that mycelium fragments into separate spores. That what we call it as arthrospores. Arthrospores I explained in my previous videos also, just to go and see. Otherwise I will provide the link in the description box to see the previous videos. And iridospores, there are budding fragmentation of a hyphae, that is arthrospores and iridospores. And conidial production, where it is common in smuts, while rust produce iridospores, also known as summer spores, that are conidial in origin and function. This is the asexual reproduction. Coming to the sexual reproduction, how it takes place sexual reproduction in the Basidiomycota. It results in the production of basidium bearing haploid basidiospores. Basidiospores is nothing but sexual spores. So how they will develop means because it has to produce the basidium, and basidia will bear the basidiospores and it release the basidiospores. Basidiospores, they are forming through sexual reproduction means what it has to takes place there means karyogamy and meiosis. Because plasmogamy already takes place, which results in the secondary mycelium, then basidiospores has to form, that is haploid spores has to form means here karyogamy and meiosis has to takes place. That is karyogamy means fusion of nuclei and meiosis means division of the nuclei. In most of the members, sex organs are not produced, and the somatic hyphae are detached somatic cells, that is arthrospores, undergo sexual process by somatogamy, that is fusion of the entire contents of the somatic cells, that is known as somatogamy. In case of Puccinia, that is rust fungi, sexual process is how it will accomplish. It will accomplish by spermatization through specialized organs called spermatia. That spermatia is nothing but their spore-like structures. They act as a male gamete, and receptive hyphae means hyphae are the receptive hyphae is nothing but specialized hyphae which can act as a female sex organs. They are ready to accept the spermatia. Here you can see in this picture. Here these are receptive hyphae, and spermatization, these spermatia are produced, and these act as a male gametes, and this is female, and they are ready to receive the male gametes, and their contents will fuse, that is plasmogamy will take place, and karyogamy and meiosis. Then it will give rise to the basidiospores. Here you can see the rust fungi. These spermatia are released, and the spermatia and received by receptive hyphae and followed by production of basidiospores. So entire sexual cycle, it involves monokaryotic phase, that is primary mycelium, and establishment of dikaryotic phase by somatogamy or spermatization of primary mycelium, and then karyogamy and meiosis in the basidium, which return to monokaryotic phase, that is basidiospores. So initially primary mycelium is there, which is monokaryotic phase, and in this monokaryotic phase somatogamy or through spermatization of the primary mycelium takes place and results in the karyogamy and meiosis in the basidium structure, and this basidium will produce the 4 basidiospores which are haploid in nature. These basidiospores will give rise to again the monokaryotic mycelium, which is known as primary mycelium. So entire cycle it rotates around the monokaryotic and dikaryotic nature. So life cycle is a alteration of monokaryotic and dikaryotic phases. So entire life cycle it involves monokaryotic and dikaryotic phases. This is the some introduction about the Basidiomycota. So just to recall what we covered in our today's session. In today's lecture we covered about the important characteristics of the Basidiomycota and how the name derived for this fungi involved in the Basidiomycota, and they produce the basidium structure on which basidiospores are produced, and some important structures and mycelium. Coming to the somatic structure, it is having primary mycelium, secondary mycelium and tertiary mycelium, and primary mycelium are compatible hyphae fuse, they will give rise to the secondary mycelium, and secondary mycelium give rise to tertiary mycelium where thick walls are hardened to give rise to the basidiocarp. And coming to some specialized structures, that is clamp connections and Dolipore septum. Clamp connections helps in the formation of new cells and division of the nuclei, and Dolipore septum which prevents the movement of nuclei between the cells and by maintaining continuity of the cytoplasmic movement. Then asexual reproduction takes place through various spores, conidia, arthrospores, uridinospores, and sexual reproduction takes place through spermatization or somatogamy. These are main things, and this entire life cycle of Basidiomycota, it is alternation of monokaryotic and dikaryotic phases. This is the today's class. In next class, I will explain about the development of Basidium, and so next class I will cover about this one. And thank you for your keen interest in this video, and remember to like and subscribe for more videos if you like this video, and please share with your friends also. I wish you a great success, health, prosperity and happiness. Thank you. One and